Hello, everyone, and welcome to today's webinar, The Irresistible Pull of Paramagnetic Beads. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckman.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box to the left and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you are having any trouble seeing or hearing the presentation. We do have multiple interactive poll questions throughout the webinar, so please make sure to participate when prompted to do so. We also have survey questions located on the far right of this page. If you could record your responses by answering the questions, scrolling down and clicking Submit at the bottom of the survey component before the conclusion of this webinar, that would be greatly appreciated. I'd like to now welcome our speaker for today's webinar, Lori Euler, Senior Manager, Drug Discovery, Genomic Solutions, Beckman Coulter Life Sciences. For a complete biography on our speaker, please visit the Biography tab on the top of the screen. Lori, you may now begin your presentation.
Great. Thank you so much. And thank you for everybody who is attending today. It's great to have you here for this little bit of an educational seminar today. As always, I'd just like to explain that this is intellectual property of Beckman Coulter. And so no copying, no distribution, et cetera, of the content involved here. We are going to start off with a little bit of a poll just to kind of level set where we all are. What methods are people currently using when they are doing nucleic acid applications? I'll give you all a few minutes to put your answers in the poll. And I'll give you a little bit of a perspective on why we asked this question and a little bit of a perspective on why I wanted to give this educational webinar today. My background is in the lab. So I spent 20 years doing research in a metabolic engineering lab. And so I did a whole host of applications that required nucleic acid extraction, cleanup, manipulation, et cetera. And I was, like most people, very married to particular technologies and particular techniques. And so that's what I use. And I will tell you honestly, beads were not part of that for me. And the main reason they were not part of it for me was because I didn't understand them. I went with what was familiar and what was common, and I didn't really have much of an opportunity to learn anything different. So for me, part of this is to really explain to you what bead-based technology is, and then give you a little bit of a walkthrough of how you can use it within your workflows. The poll results are pretty interesting to me, actually. We do have quite a mixture of, quite a bit of folks who use a mixture of both column and bead-based applications. So that's really great. Some of you might already know some of this, but it's always a little bit helpful to understand the background on it as well. So moving forward, this is a really simplified version of nucleic acid purification, and it's really to level set a little bit on some terminology. So there's really two big paths to getting purified nucleic acid. The first is you're going to start with biological material. You do some lysis of that material, break that cell open. You need to get that nucleic acid out of there. So you'll start your process with your lysate. A lot of times that is quite dirty, quite crude. You will have cellular debris in there. You oftentimes can still have some carryover of sample buffer, things like that. So that is in our world, and what we'll be talking about down further in this presentation, that's extraction. If you're starting with an in vitro reaction, so say you have an enzymatic reaction, you have something going on in your lab, but you need to do that purification of nucleic acid. You'll do your in vitro reaction, you will have a crude product in there, and you're going to do cleanup. So this is just level setting you a little bit more on the difference between extraction and cleanup. Extraction is generally a bit more complicated, again, because there's a lot more in that crude lysate that we need to get away from that nucleic acid of interest. They're dirtier samples in general, so a little bit more complicated than just a simple cleanup step. So I mentioned before, I was very married to certain technologies. As a grad student, phenol chloroform extraction was kind of the standard. This is one of the older technologies that a lot of people are very familiar with, not so much in fashion anymore. And the main reason for that is because of the toxic compounds that you're using. So getting away from using something like phenylchloroform is really important. One of the other reasons that folks want to get away from things like using chemical based purification methods, not just the toxic compounds, but it's not really scalable. It's a bit labor intensive. You have to be a little bit precise when you're removing that aqueous phase. And so not really amenable to scaling up, not amenable to a large number of samples. What most people are familiar with nowadays are solid phase technologies. I think column based methods are the thing that most people are familiar with. So, silica membrane based technologies, typical columns that spin columns that people are used to, spin columns that can also be used in vacuum manifolds, and filter plates. I think this is what most people are most familiar with. You will have DNA binding onto that silica gel membrane. In certain situations, it's all buffer dependent on what actually binds to that membrane. But the method that we're going to talk about today are magnetic beads. So again, solid phase because you are binding your nucleic acid to the surface of the beads. Some similar principles with the wash and release, but different solid phase that you're utilizing in bead-based technology. So we call this SPRI technology. And just so that everyone's on the same page, solid phase reverse immobilization. Reverse and immobilization are kind of the two keys in this phrase here. On the left, what you'll see is that you have the solid phase, so that's the bead itself, and this kind of illustrates that immobilization part of it. So you are going to immobilize your nucleic acid of interest onto that solid phase. This is all selective binding, and it's also specific binding of nucleic acids. This is what allows those contaminants to be washed away because you're only binding your nucleic acid. The reason that you need this to be reversible is because then you need to actually get that nucleic acid out off of the bead into solution and recoverable. The purity of your sample is really dependent on that selective binding. So again, you have selective binding so that it's just nucleic acids. You don't have proteins, crude parts of that crude extract in there. The yield is dependent on the binding and then the release itself. So how much you're going to get at the end of this process depends on how well that binding happens and then how well that reversible binding is. So again, you need to be able to release the DNA or the RNA from that bead. And so this is just a little bit of an illustration of solid phase reverse immobilization and what we mean by that. I'm going to give you a real brief history of this technology. So this was developed in the 1990s at the Whitehead Institute. This was really a pivotal part of the Human Genome Project. So it was used to clean up PCR products for Sanger sequencing. About 2000, Agincourt Biosciences was established, and the whole point behind Agincourt was to commercialize this technology. In 2005, they were acquired by Beckman Coulter. So a lot of times you still might see folks refer to these as Agincourt beads, things like that. So you'll see a little bit of both tech terminology, but Agincourt and this SPRI technology is part of Beckman Coulter Life Sciences now. So let's talk a little bit about these SPRI reagents that Beckman offers. There's three main components here. The first, obviously, the magnetic beads. This is that solid support for the nucleic acid binding. Again, without that solid support, there's nothing for the nucleic acid to bind to. There's also a crowding reagent. And essentially what that crowding reagent does is it really squeezes the nucleic acids out of solution and it drives them to bind to the beads. Concentration of that crowding reagent is really critical. It has a lot to do with the size selection, and so we'll talk a little bit later about how ratios of the reagents can really dictate exactly what you get out of your reactions. And then the binding buffer. This is really important. The binding buffers themselves contain salt ions, and this, we help adjust the pH for binding with this binding buffer. The cation that you see here acts as a bridge between the bead and the nucleic acid that's in the sample itself. pH can really impact this process. And so it's really important to be mindful of things like that. So if you get carryover of any sort of sample buffers into this part of your process, you have to be really mindful of the pH. You can really decrease your binding efficiency if the pH isn't optimal at this stage. So let's look at the beads a little bit more because this is really the magic behind this technology. And you can see on the left-hand side, this is truly the components of the bead itself. There is a carboxyl-coated surface for nucleic acid binding. So that is what the outside of the bead is. It's this carboxyl-coated surface. Um, Inside, in this inner layer, you have the magnetic layer. This is crucial because this is how you manipulate beads during the workflow. So we have to have some way for these beads to move around, for us to be able to remove contaminants, et cetera. And it's that magnetic layer in that inner core that really is helpful for that. And then finally, the core of the bead itself is polystyrene. The reason for this is for buoyancy. You don't want these to just sink to the bottom. If you do, then you've really not, you don't have the optimum interaction with the nucleic acids. So the buoyancy is really key. Buoyancy also plays a huge role in how fast this works. And in, it's really a tight interaction between the buoyancy and then the magnetic efficiency and the magnetic strength as you're going through this process. Let's look at the surface a little bit of these beads. You can see on the right-hand side, there's a picture of what looks like cauliflower. It is not. It is the topography of the beads. The reason that these are not soft, smooth beads is that by having this intricate topography, it gives us a higher surface area. And with that higher surface area, we get maximum binding of nucleic acids to these beads. One of the other things that is really important is the size of the bead. So these are optimized for performance, both again for interactions within solution, interactions with the nucleic acid, their attraction to the actual magnets that we're using. So the size is really optimized both for recovery and for the manipulation of the beads themselves. One of the great things about the Beckman SPRI technology and the Beckman beads is there's really tight control over the manufacturing of the beads. So there's really tight control over things like size, things like the surface area. Um, a hallmark of a successful extraction or cleanup is really for it to be reproducible. So you want to make sure this is not the most exciting thing you're doing in your lab. This is not what you want to troubleshoot or what you want to overthink. So by having that really highly controlled size, highly controlled surface area, it really allows you to get reproducible results every time you use these beads. This is a really generalized look at what SPRI methodology is. And so I kind of introduced by saying we have extraction, we have cleanup. Regardless of what you're doing, this is really the basics of SPRI technology. So you have a bind step, you have a wash step, and you have an elute step. Binding, it's really simple. In the presence of our proprietary buffers, your nucleic acid is going to bind to the bead surface. And so again, this is very standard methodology, regardless of what your sample type is or what kit you're using. It's just those buffers that are kind of the nuance there. Washing is really important. You can do multiple washes with this sort of technology, which is really great. Your beads are going to be immobilized with the magnet. So they're kind of drawn out to the side of the tube. It allows really easy removal of the liquid in the middle. Again, your contaminants. You can do multiple wash steps to make sure that you're really getting all of those contaminants out of your solution. And then finally, elute. You have to be able to get your sample out at the end of this process. Add a buffer, DNA is released. The beads are still immobilized onto the side, so they're still attracted to the magnet. But because you've changed the environment with the buffer that you're using, the DNA is no longer immobilized on those beads. So your DNA or your nucleic acids are now in solution and they're easily removed. This is a really simple process. So the other really great feature of this is it's easily automatable. This is something that you can do manually on your lab bench. You can do one-offs on your lab bench, depending on the type of magnet that you choose. But this is really amenable to being automated on things like a Biamech system. If you are doing multiple samples at a time, you can do up to 4 96 well plates at a time with these assays. So makes it really easy for you to automate processes. We're going to launch another poll question here, really just to make sure everybody's on the same page, everybody's understanding what these beads are about. So it's a little bit of a quiz, I would say. So which of the following does not promote nucleic acid binding to the SPRI beads? We have a couple of responses coming in. And so it's the magnetic layer. You guys are great. The magnetic layer does not promote the nucleic acid binding to the SPRI beads. The magnetic layer itself is what allows you to manipulate the beads throughout the process. So great job, everybody. Gold star. Everybody understood that. Magnetic layer is not for binding. I'm going to show you a quick little video just to kind of give you an idea of what the process looks like. And let me make sure this plays for you. Okay, so this next slide is really a time-lapse video to show you what this actually looks like. And I mentioned earlier that buoyancy plays a really big role in this. That's the whole reason we have that polystyrene core, is because one, we don't want the beads to just sink to the bottom before we apply sample or anything like that. We don't want to have constant motion. So the beads, the buoyancy is really key. So the beads are not going to sink to the bottom. This allows really that time for the interaction, that time for the binding of the nucleic acid to the beads. But one of the hallmarks of SPRI technology is it is a nice, efficient, time sensitive process. So you're not going to take a long time to get the binding to happen. It doesn't take a long time for you to then use the magnet to then immobilize, to pull the beads to the side to continue your process. So this is real time immobilization on the beads. What you're seeing here is you have a magnet at the base of the plate where these samples are. And as that magnet is applied, the beads are going to accumulate at the base. So again, the speed is dependent on the strength of the magnet. So that is pretty crucial. And then again, how buoyant the beads are. So the more buoyant, the slower this is actually going to happen. But you can see in this time-lapse video, it's a pretty overall fast process. The other thing to consider too is that the overall viscosity is going to play a role. So we talked again about how important it really is, the sample that you're putting into this reaction, the sample that you're using to apply your beads comes to viscosity will play some role in this. This is just kind of a top-down view. I think, it's sometimes really hard to think through what it's going to look like in a 96 well plate. Again, this is a top-down view. And this is if we have a magnet where the magnet is around the outside of the wells itself. So what you're seeing here is that you get these beads in these really tight rings. So that dark circle on the outside is actually the SPRI beads. The reason I want to show this is to show you how easy it really is to pipette, if you're doing this manually, the liquid from the center of the well. So you're not going to disrupt these beads. They're strongly attracted to the magnets on the outside, and it gives you that whole center of the well itself to remove your sample. One of the reasons I really like this visual as well is some customers really have this idea that this is a really more difficult technology, right? So this is something that people say, well, I'm an academic lab. I have turnover of students in my lab. I have turnover of technicians, and I need something really simple. So they think that, hey, a column is going to be the simplest thing for them. I think this is a great visual to say you may not understand beads yet. You may not, may just think because it's different, it's going to be a lot harder. It's really a simple pipetting step based on, having that open center and being able to remove the contaminants, do your wash steps, and then to elute and remove your sample. So I always like to show this when I get that argument that, well, we have a lot of turnover. This is going to involve training of associates, things like that. It's really not. Once it's something that you establish in your lab, you have the magnets on hand. It's really not any more difficult than anything else. For me, one of the big advantages is not having to rely on a centrifuge, right? So if you're doing a 96 well plate, and you have to rely on a centrifuge to spin those plates, if you're doing a silica-based method, that's a lot more labor intensive, a lot more hands-on time, a lot more opportunity to maybe have a pause in your workflow if you have shared equipment in your lab, and everybody needs that centrifuge today. So I always like to dispute the argument that this is really a lot more complicated to set up in a lab where you may not have highly skilled technicians, or you may have a lot of turnover in your lab. And again, the other thing is for scientists in a lab, this is not the process that they want to think about. So having an easy process like this really allows you to do this fast and move on to the important things. These are some magnets that are very automation friendly. So I just wanted to give an idea of what these looked like as well. So as I mentioned, and kind of if you remember the one we just showed, that's that ring style. This is really the preferable type. They tend to work the best, and people find it much easier because, again, easy removal from the center of that well to remove the contaminants during the wash step, and then for the final elution step. On the top left-hand side, though, you will see a different type. So this is a bar magnet. The only difference here is that you're then going to get your beads all accumulating on one side. So if you're doing this by hand and you're going to pipette and you have a bar type magnet, you're just going on the side that the magnet is not. If you're using this in an automation format, it's just a simple tweak to the automation script to say, hey, we're going to pipette away from that magnet. So a little bit about the kits themselves. There are certain things that are pretty standard in our kits. They, will provide buffers like the lysis buffer that you need. Again, if you're doing an extraction, you have to break those cells open. So you'll have a lysis buffer in there. There are nucleic acid digestion steps as well. So for instance, if you are looking to do a DNA extraction, you obviously don't want your RNA to be hanging around as well. So there are certain steps in here when you can do things like an RNase digestion. So you can get rid of the nucleic acid that you do not want. There are rebinding buffers in here. There's also this allows you to remove contaminants because of all of these buffers. So a little bit about what makes us unique. This is a little bit of a summary. So again, these paramagnetic particles, they're easily automated. So again, this is really great. And one of the huge benefits over other methods is that you can automate this process quite easily with the same beads, same kits that you've used throughout your whole process. I think, a lot of times people say, it's okay, I can use the column and then I can go up to a filter plate. There's really not a huge there's not a great correlation, a linear correlation between that. You're always going to see some differences when you do automate or when you miniaturize a process. But again, these are easily automatable, easily scalable. You don't need that centrifuge, save for the lysis step. Obviously, that lysate clearing can require a centrifuge, and you don't need filtration. Again, that uniform bead size is really important. Highly reproducible results is key. You don't want to one day go in with a sample, think you've have everything worked out, go in the next day and get half the amount of nucleic acid out of it. It's going to slow your work down. It's going to really prevent you from moving forward and using it in downstream applications. This is, to me, is a big thing as well. So we can show you yields, we can show you purity. If you can't use things in downstream applications, then there's no point in going through the process, right? Nobody ever really stops at just isolating the nucleic acid. It's always going to be used for something else. So because we're not eluting with salts, what you're eluting in is really amenable to immediately going into downstream applications. There's no buffer exchange needed or anything like that. Again, low non-specific binding is really key. So that polymer surface is important. You don't want to have binding of any of the contaminants, particularly when you're doing extraction. So you don't want any carryover of those contaminants. You don't want to pull some protein along with you. I think a lot of times people who use column-based methods find that it's really easy for them to overload their column, and so they get flow through of contaminants. I think using a bead-based technology is one really great way to prevent that carryover of contaminants that is really a big problem that plagues column-based technologies. That high surface area to mass ratio, this really increases the binding capacity. Again, I think there's a lot of misinformation out there about, you could get more if you use a column because you can do multiple binding steps, etc. You're still going to hit a certain point on a column-based technology where you're not going to bind anymore. But that high surface area of these beads really allows an increased binding capacity. Fast magnetic response time means rapid purification. So we talked about the buoyancy and the magnet strength and how important that really is. Those factors really allow these rapid purifications. It allows you to get through this end of your workflow fast, move on to the more important stages. Again, because these particles are size optimized, they're really great for automation and for automated applications. Not having that variability means you set up your automation and you don't have to tweak it down the road. I think Beckman has really high quality standards. Our internal QC is quite extensive, so we are not going to have huge variation in bead size, which would maybe affect how your automation is working downstream. So I'm not going to run you through every single kit that we have. I just wanted to give you an idea of what our kits are, extraction versus cleanup, and then really give you an idea of how you can find these things and how Beckman can kind of help you understand what kit is right for you. So on our website, we have it kind of laid out pretty easily for you. If you're looking to do DNA extraction, you can find our sample prep page that outlines your DNA extraction solutions. And I think one of my favorite things about this is you can look at this based on what input material you have, what your output is, and then we'll tell you what are some of the suggested applications. So this is a really great way to say, I know I need to do DNA extraction. I have FFPE tissue. I'm not really sure what kit I need. I just know Beckman has a ton of DNA extraction kits. Charts like this that are on our website help you pick exactly what you need. So in that instance, you would be really, it would be best for you to use a Formapure XL DNA kit. One of our really unique kits, this is kind of one of my favorite things, is if you have FFPE tissue and you need to do DNA and RNA, we have a Formapure XL Total Kit. The reason this is really a great benefit is if you do need RNA and DNA, if you have FFPE tissue and you start your process for extraction, and then you need to split that sample into two to then go down two separate pathways to get your RNA and then your DNA, you're really essentially starting with half of your input And so you're going to get less out of that. So having one kit, one single chemistry solution for the isolation of RNA and DNA in these FFPE samples means you're not splitting it, and you get much better yield, better results with using something like that XL Total kit. We also have RNA extraction solutions though. So again, you look based on your input material and based on your output. So same sample types in a lot of instances, same input material, but you're looking at your output now. So you want RNA instead of DNA. Form a pure XL RNA kit is the one to go to. Again, if you're not looking for both in the same sample. And then finally, our cleanup kits and our size selection reagents. I think most people are probably very familiar with our Ampere XP, sort of the gold standard in the industry, recommended in over 200 NGS cleanup kits, or in cleanup for for NGS kits. So again, if you look for your input material, what do you have? We have PCR products, and we're looking for just to clean it up for clean up that DNA. This leads you right to our Ampere kits. So another poll question for everybody. Of those two techniques, extraction and cleanup, which one do you do most often, or is it sort of equal? Do you use both in your lab all the time? I'm going to say my prediction is that people probably use both techniques equally. I think especially folks that are in a cloning workflow, gene editing workflow, things like that. Both techniques are really important. Thinking through extraction, though, there are, the nuances. It's a lot more complicated with the nuances with sample types. So yeah, I think my prediction was equal. And as I look at the results, it looks like people are equally doing cleanup and extraction in their lab, but there's also a lot of folks doing, doing extraction more often than anything else. Okay, so I'm going to run through a couple of case studies just to give you a little bit of an idea of how these kits really do fit into workflow, how they're used. For me, it's great to hear somebody talk about a product and how great it is, but seeing the data behind it is really critical. And if there is, if there are all of the case studies that are presented here also have supporting data on our website, so you can find either scientific posters or application notes that will give you the full picture of the case studies that we're going to talk through right now. So first case study is with the Ampere XP reagent. The impact of cleanup kits on sequencing quality is really key. This to me is one of those perfect examples of garbage in, garbage out. And so cleanup before NGS is really quite pivotal. If you don't input enough sample or a high quality sample into your NGS workflow, you're going to see that reflected in the results that you get. If we look at the percent recovery, for optimal quality and efficiency of your NGS downstream, your percent yield should be between about 80 and 90%. And so if you look at Ampere and you compare it to some of our competitors, we're on par or better than our competitors, much better than one of the competitors that you see on this table. But you will achieve that 80 to 90% optimal recovery with the Ampere kits. I think again, I've said before, this is not the most important thing you're doing in your lab. This is not what you want to spend your time on. This is not what you want to troubleshoot. So hands-on time is really important, and reducing that hands-on time is key. So your time is your most valuable resource. It's what you don't, and again, this is not what you want to spend your time on. If you look at the chart on the right-hand side, the graph on the right-hand side, it's a comparison in individual cleanup step time between Ampere and two competitors. So Ampere is sitting just under about 30 minutes for one cleanup step. Competitors are up to 45 minutes for those cleanup steps. In the majority of these library construction kits, there are multiple cleanup steps that are needed. So if you were only doing one step, only one cleanup step, it's about a 15-minute time savings. But as you need multiple cleanup steps in this library construction, you're going to amplify that time savings. So if you have two cleanup steps, you're now looking at 30 minutes difference in the hands-on time that's required using Ampere versus a competitor. And then Predictable performance. This is really what people are most concerned about, right? So you got your, we know that we're going to get yield. And again, some competitors are okay with yield as well. But uniformity of the DNA fragment size, it's going to affect the quality and it's going to affect the efficiency of your DNA sequencing. You really have to consider that initial fragment size. It has to be correctly targeted because you're going to have additional base pair length as you put adapters and things like that onto your fragment for sequencing. So you really have to optimize that initial fragment size. And again, fragment size concentrations should remain high and predictable with different binding ratios. So if you look on the right-hand side, you have your genomic DNA on the top left. You have a sample using Ampere XP on the right. We have some competitors on the bottom. And again, the key here is as you change those ratios, as you're looking to isolate different size, if you're doing size selection, you really want to have that same concentration. You don't want to lose sample as you're changing those ratios, as you're looking for different fragments. You want to have that uniformity of DNA fragment size, regardless of the ratios that you're using, and that's really going to increase the quality and the efficiency of your sequencing downstream. Okay, I'm going to talk through a little bit about two case studies with our extraction reagents. There's really been quite a boom in research, cancer research involving FFPE samples, but especially with cfDNA samples or cell-free DNA samples. Analysis of cell-free DNA for cancer detection, for prognosis, for patient monitoring has really significantly improved clinical management and outcomes for patients. The primary challenge, though, when we're talking about cfDNA samples, we're talking about liquid biopsy techniques, is that cfDNA is typically found at extremely low concentrations in bodily fluids. So we're talking a range of about 1 to 50 nanograms of cell-free DNA if we take 1 ml of plasma from a sample. So again, really small amount of sample involved here when we're looking at this cell-free DNA. Of the total cell-free DNA found in plasma, fragments that are originating from tumors, so tumor DNA is probably about 0.01%. So again, you have a really low occurrence of the cell-free DNA, and of that cell-free DNA, there's a really small fragment of that is originating from tumors in cancer patient samples. So what this really translates to is that you really have to have efficient techniques for cell-free DNA extraction. This is really essential for tumor detection in precision medicine applications. I think most people are very familiar with using FFPE samples in cancer research. Again, there's a lot of pitfalls with the sample type. It's not a sample type that is easily manipulated. It's quite harsh treatment to actually get those FFPE samples preserved. So there has to be really efficient techniques to get the nucleic acids out of those samples as well. So if we look at this first slide, this is really a representation of the nucleic acid extraction from some FFPE tissue samples. On your left-hand side, you're seeing the nucleic acid yield. In general, if you compare FFPE samples from different sample types, so you can see there's breast, lung, intestine, and prostate samples, we've compared the PharmaPure XL kit with a column-based method. So if we look at our comparison with the column-based method, overall, we're about the same, right? So we're on par with them in several instances. Our yield itself is much better. The prostate samples are a little bit of an outlier in that yield, but again, we're on par with yields, better in most instances. One of the really important things, though, as I mentioned, these are not gently treated samples. So what we need to ensure is that those samples can actually be used for something. So RNA integrity is a really great way to look at, hey, can I actually do something with these samples? So if you look on the right-hand side and you look at RNA integrity, you see a much different picture. So we're not on par with our competitors at this point. We're much better. So the RNA integrity from all sample types, from breast, lung, intestine, and prostate, is much better using the PharmaPure XL kit versus the column-based methods. And so again, having that increased integrity of your nucleic acids, it really supports improved sensitivity of things like qPCR or NGS, any of those downstream applications that you're going to take these samples and use them for. Again, yield is great. You need to have enough sample to do something with it downstream, but it has to be of a high enough quality that you can actually do the assays downstream. This is a little bit of data on cell-free DNA extraction from plasmid samples. So again, remember, the abundance of this cell-free DNA is extremely small in plasma samples from patients. So for this study, we asked an independent investigator to compare our Apostle Minimax kit with a QIAGEN circulating nucleic acid kit. And in all parameters measured, the Apostle really significantly outperformed the QIAGEN kit. For this experiment, blood was collected in EDTA tubes, plasma was isolated. The cfDNA was isolated using the kits I just mentioned, so the Apostle Minimax from Beckman versus a QIAGEN circulating nucleic acid kit. The researcher did quantification using a qubit, and then we also did digital droplet or ddPCR with primers that targeted some housekeeping genes, so actin gamma and DNAJ heat shock proteins. So if we look at the concentrations that we isolated using both kits, you can see that there's a 34.3% higher yield using the Apostle kit. So again, we're talking really small amounts of cell-free DNA in these samples. So getting that higher yield, that 34% higher yield, is quite significant. When you're looking at these specific genes, so we're looking at this middle graph here, we're looking at those specific genes that we did the ddPCR assays on. You can see that there's a 15% higher yield with the Apostle, which are those pink bars. And then if you look at these cell-free DNA fragments with a bioanalyzer, we can see that those mononucleosome cfDNA concentration in these Apostle samples was 70% higher. So if you kind of look at this as an overall, there's about a 40% average yield increase for all parameters using Apostle versus the QIAamp kits. I'm going to tie these two together a little bit and present you a little bit of data if we want to look at cfDNA and FFPE samples kind of in concert. And we're going to look at mutation detection in these cfDNA and FFPE samples. The other thing I want to point out with this is that the extractions and all of the library construction were all done automated on a Biamech workstation. So again, this is just a little bit more credence to the fact that these are easily automatable and it's amenable to a lot of different scales of workflows. So you can see in the table on the right, the sequence coverage, and this is what's really important, is that all samples had sequence coverage of 50x, at least 92% of the bases sequenced. These two charts really explain to you that the identification of variants in both sample types. So we wanted to look for single nucleotide variants as well as insertions and deletions in the sequencing results that we had gotten from those samples. On the left, you can see that we looked at the single nucleotide variants found in both sample types. In the majority of these sample types, we found both. So there was really no statistically significant difference between identifying the single nucleotide variants if we used a cfDNA sample or an FFPE sample. When we look at the the insertion and deletion mutations, story is a little bit different. There is a bit of a statistical difference. You can see in these kind of purplish bars is where we see only in that sample type did we find that insertion or deletion. So there is a statistical difference. More indels were identified when sequencing cfDNA versus the FFPE samples. And one of the other things is, again, what do you do with this, right? So what do you do with this information? So we wanted to determine if we were able to take this data and find any pathologically relevant variants. And so to do this, we compared what we had seen in the sequencing results against a list of variants from the NCBI ClinVar database. We only used variants that had a clinical significance, uncertain significance, likely pathogenic. We did this for 4 different tumor types, and we were able to identify 2 variants from the database that we could only find actually using the DNA from the FFPE tissue. So when we, again, looking at this FFPE tissue, we were able to find certain associations these mutations had with clinical pathology. Um, in most of our samples, though, we were really unable to find any variants that were identified with things in the database. However, we were able to find variants at the same position as variants that are in the database. So this could alter encoded proteins, could be candidates for further study. So these are just things that may say to a researcher, okay, we don't know the clinical variants of this yet, but this could be something that could be important for these patients, or something that really warrants some further investigation. And just a little bit of summary of those 2 case studies. So when sequencing of cfDNA, we captured the majority of the variants found in that sequenced FFPDNA. We did see a difference in indel identification using cfDNA versus FFPE, and that was pretty specific to one tissue type. And we have identified some clinically relevant variants, but also we were able to find some things that could potentially be areas of investigation in the future. So I did mention that that last set of data was generated in an automated fashion. So the extractions as well as the library preparation was all done with a Biomech. Just want to give a little more information on Biomech automated solutions for any of our genomic applications. You can see at the top here, kind of a little bit of a workflow feel to this graphic. There are so many steps within your genomic workflow, within a cloning workflow, that really require extractions and cleanup steps. And so all of these are really amenable, again, with SPRI technology, with SPRI kits. They're very amenable to high throughput automation. One of the benefits that Beckman brings to the table is our genomics and our automation teams are part of a biotech workflow solution team. And so our whole goal is to work with customers to say, what are your research goals? What do you need to get out of this? What's your workflow look like? How can we help you automate that? So I think it's really powerful. It's a powerful comment that we have these 2 teams together. We can really help you do things like improve library quality. There's quick implementation. Again, there are methods that exist, so we can quickly implement these on your Biomech so you can get a faster time to results. And again, there's really a huge portfolio of kits that can be automated on these Biomech workflows, Biomech systems. Again, this is a bit of an eye chart. I am not going to read this to you, but I just wanted to again point out that if you're doing things like NGS library construction, on the Beckman website, we have a really easy to use chart that says what kits you might use, if what particular NGS library construction methods you're using, and whether or not we have demonstrated methods for that. So this is really an easy way for you to say, okay, here's what I need to do in my lab. Can Beckman help me with that solution? And again, there's also a very similar chart if you're doing nucleic acid sample preparation. So again, here's my sample. Here's what I want to get out of it, RNA or DNA. Beckman has the kit. Can I automate it? And I know a lot of times the kits that you buy off the shelf, the protocols that we have on a Biomech aren't always going to fit exactly what you're doing. I think one of the really strong offerings that Beckman has as well is the ability to work with our proof of principle team to help you develop some custom solutions. So, this isn't brand new technology and brand new kits. It's taking what's already available and saying, how do we help tweak that to optimize extractions, to optimize what it is that you're trying to do in your lab? There's a list of some of the past projects that we've worked on. The really great thing is once we do this with a customer and they say, hey, this is great, it worked, here's some data, we then generate app notes, we generate supplemental protocols, things like that, and we put them all on our website. So you may, very easily go in and say, hey, this is great. This is the kit I want. You get it in your hands. It wasn't quite didn't quite give you the results that you wanted. Maybe your sample is a little bit unique. Maybe your sample doesn't really fit the parameters of any of the ones that we list in our input documents. That's where our proof of principle support team really comes into play. So you can easily find contacts for us on our genomics page on Beckman.com. And you can also find any of the supplemental protocols that we may have done with other customers. So, don't just look at the standard IFUs for a kit. Take a peek around these proof of principle pages to see if there are any custom protocols that may fit your workflow a bit better. And with that, I want to thank you again for taking some time with me today. I appreciate you attending the webinar. I'm going to bring Cassie back on. Let me know if there are any questions from the group and if there's anything I can address.
Thank you, Lori, for your informative presentation. We will now start the live Q&A portion of the webinar. So if you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many questions as we have time for today. Let's go ahead and get started. Our first question is, why are paramagnetic beads not ferromagnetic, and what is the advantage?
So these are paramagnetic beads because for us, we found that having the ability to have them free floating even when they're in the presence of the magnet, and then only have that magnetic pull when it's applied to the magnet, works much better for these technologies. So this whole technology was based on a paramagnetic bead type principle. Again, as I mentioned, these were developed many years ago in the 90s for Sanger sequencing, PCR cleanup. And so this is what was found to really be the best back then.
Wonderful, thank you. Our next question here is, does your extraction for cfDNA distinguish between exosomal DNA and truly free DNA?
Yes. And so, as I mentioned, the data that was generated that was in those case studies is from an application note that is available on our website. There's a lot more data in that. I only picked out some bits and pieces of it. But yeah, there's a little bit more data on the how you can distinguish between exosomal DNA and truly circulating cell-free DNA.
Okay. Our next question here, is there a size selection discrimination possible with the beads?
Yes, absolutely. So our SPRI select beads, that's exactly their purpose for them. So size selection. I kind of briefly mentioned doing size selection with different bead ratios, things like that. So yes, you can definitely do size selection within your NGS cleanup workflow using something like a SPRI select bead. There are some really great pieces, again, going back to check our website, but there are some really great pieces on there that walk you through what size selection really is, how you do it, how the ratios are important. I think, a lot of times we have competitors who say that their beads are kind of a drop-in replacement for the Beckman beads. It's not always the case because a lot of times the ratios, so they'll show the same data, you're still getting the same size selection, et cetera. But the big difference is a lot of times there's a lot more there's a lot of more tweaking of the ratios that need to be done. So a lot of times you're using twice the amount of beads, twice the amount of the reagents as what you would with a Beckman SPRI technology bead. So yeah, on the website, there's definitely some things that can walk you through as well, how to do size selection with these beads.
Great, thank you. Our next question is, does SPRI technology have an upper limit on nucleic acid size?
Yeah, so I know this is a question that comes up a lot too, because there's a lot of folks that are looking to do, long read, things like that, and they want larger fragments. There are pieces of literature out there that show folks using these. It's definitely something that we're interested in. We're always, again, the ability of having this proof of principle team to help you tweak things is great. It's also really helpful for us to understand if there are other things that you need to do. So if you need to have a larger fragment size, this is something that we want to work on with customers as well and understand how important that is to them and what applications they're using it for. I can see the question, so I'm just gonna jump in. There's one asking about various protocols for washing. Should we wash on or off the magnet? Should we resuspend or just let the beads sit in the buffer? So again, I think this is one of the aspects that people tweak a lot, and one of the things that our POP team sometimes gets questions on, and we help with that. Um, I think there are, again, it's going to depend on the kit that you're using. But there are some instances where, yes, you will wash on the magnet. It's much easier. And there are some instances where customers will want to do that wash off of the magnet so that the beads themselves, again, are in suspension. And should we resuspend or just let the beads sit and buffer? Again, this is kind of going back to that buoyancy issue again. The beads are not going to just randomly sink to the bottom of your tube or your plate really fast. But it's really important when you're doing any of these assays, especially from the start, to have really good resuspension of your beads. So, if they're sitting around a long time, if your bottle of beads is sitting around, you are going to see some settling. It's just what happens. So again, when you're going to do your assays, make sure you have them resuspended really well. And then as you go through your protocols, your sample type might be a little bit different. Again, we mentioned how viscosity might be different, how the magnetic strength might be different. There could be instances where you might need to tweak a bit if you resuspend it, how long you need to leave them sit in resuspension, things like that.
Great. Thank you, Lori. Our next question here, do you have protocols for preparation of RNA and DNA separately from the same biological sample?
Yes, we do. So again, it's going to depend on what your starting sample is. I mentioned the one instance of Formapure. Formapure, we have a DNA kit, an RNA kit, or a total kit where it's both. So yeah, there are certain instances. We do have kits for RNA alone and for DNA alone, and a lot of times the input samples are the same. So in some of those instances, and again, if I'm understanding the question correctly, in some of those instances, yeah, you are going to have to you will split your sample, but we do have kits that work on the same sample type.
Okay, thank you. Our next question, can you tell us more about the SPRI beads and their use for size selection, and what's the difference with the AMP-XP beads?
So this is a great question. We hear this a lot from customers. A lot of times customers say, well, I use both for both purposes. Um, the biggest difference with the two is really our Ampere XP beads are really used for PCR cleanup. And so that's what that's what we have um, quality metrics around using them for. The SPRI Select kit. So again, this is all based on SPRI bead technology, but we have a SPRI Select bead that is used for size selection. And so the technology itself is the same, the components of the bead the same. It's just that we will use the SPRI Select beads for size selection versus the Ampere beads, which is really favored for things like PCR cleanup.
Okay, great. Thank you. So our next question that we have here is, can I interchange Ampere XP beads and SPRI beads in cleanup?
So again, I'm gonna assume this question is the SPRI Select beads. Because the Ampere beads themselves are all, they're based on SPRI bead technology. So again, if you're thinking through cleanup, it depends on what your ultimate goal is. So again, if you're doing PCR and what your input is. So if you're doing PCR cleanup to use downstream in NGS workflows, so if you're thinking about it in that NGS cleanup step, yeah, Ampere XP beads are great. If you need to do size selection, then the SPRI Select beads are your go-to.
Fantastic. And it looks like we have time for just one more question. What are the benefits of automating SPRI reagents? If I'm interested in doing this, how do I get started?
So I think there are tons of benefits of automating SPRI reagents. Again, I think one of the biggest things is that throughput. These are things that, again, this is not what you want to be thinking about in your lab. It's not what you want to be spending time on. There's a lot of labor costs involved in doing these techniques manually. So automating them is great. If you are interested in figuring out how to do this, on our website, there are various ways to get in touch with us. There are some form fills that you can do. You'll be getting the opportunity after this webinar as well to reach out to us. And we're happy to get you connected with our application scientists who can help you start to understand what kits and exactly how to automate them.
Great. Thank you, Lori. Did you have any final comments for our audience today?
No, just again, thank you all for taking some time with us today. Please reach out if we can be of any help. If there's any kits you want some more information about, if there's automation that you want to talk through with us, we're happy to help.
Thank you again, Lori, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions that we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. And until next time, goodbye.